Hello everyone, my name is Paulina. I am a PhD student in Biomedical Sciences at the Universidad Nacional Autónoma de México and the Instituto Nacional de Medicina Genómica, also in Mexico. Today I'll be talking about how changes in gene expression patterns reveal reorganizations in network models associated with late-onset Alzheimer's disease. So, late-onset Alzheimer's disease, or LOAD, is the most common cause of dementia worldwide, and it is a complex disorder. The diverse phenotypes associated with LOAD result from interactions between genetic and regulatory factors that influence multiple biological pathways throughout the disease progression. To broaden our comprehension of transcriptional modification in late-onset Alzheimer's disease, in the dorsolateral prefrontal cortex, or DLPFC, a brain region located in the medial frontal gyrus of humans that has higher order cognitive functions, we employed a network-based strategy to build a gene co-expression model, which we analyzed at the global, local, and mesoscopic scales. So we obtained raw bulk RNA-seq matrices from the Religious Order Study and Roche Memory and Gene Project, or ROSMAP, from the DLPFC. The quality control and normalization of data was performed using the NOISEQ and the RSINE packages in R. We classified patients into control and aided groups according to neuropathological changes, following the National Institute on Aging and Alzheimer's Association criteria. Data was stratified by pathological diagnosis, resulting in two expression matrices that were used to build the co-expression network models based on mutual information. We used mutual information because it not only measures linear but also non-linear associations. This feature is advantageous since many relationships in the transcriptional phenomena do not follow a linear statistical dependence. Then the downstream analysis included, as I said before, the global, mesoscopic, local, and therefore the biological analysis. To analyze the networks at the global level, we perform a topological analysis that includes calculation of global metrics such as global centrality, associativity, and global transitivity, or clustering coefficients. Here we observe two networks with similar overall structures. However, only 68.39% of the links were shared between the two networks, indicating difference beyond global topology. And structural divergences became more evident at the mesoscopic and local scales, as we are going to see. Local level analysis focused on gene centrality differences. We identified genes with differential centrality based on degree and betweenness measures. First, the degree of a node in a network is the number of connections it has with other nodes. Based on degree, genes that were central only in the control network were enriched for biological processes related to small nuclear RNA binding and epidermal structure. In contrast, genes that were uniquely central in the LOAD network did not show significant enrichment for any biological process in the hypergeometric test. We also identified a set of 83 genes that were consistently co-expressed with all AD-specific hubs and were exclusively first neighbor of these hubs in the AD network. Functional enrichment analysis of these newly associated networks revealed four main functional groups: cell structure and scaffolding, immune response, chromatin modulation, and microRNA-mediated regulation of transcription. Based on betweenness centrality, a measure of centrality that quantifies the frequency of number of times a node is between the shortest paths of other actors, we found that 39 genes lost their high centrality status in the AD network. These genes were associated with three single-nucleus RNP spliceosomal complex assembly and skin barrier formation. Conversely, 44 genes that were not central in the control network gained high betweenness in the AD network. These LOAD-specific high betweenness genes are primarily associated with synaptic function-related process. At the mesoscopic level, we identify gene models and their biological functions. The AD network exhibited higher modularity but reduced integration between gene communities, suggesting more insulated transcriptional modules with weaker crosstalk. A gene model similarity matrix revealed that while 10 models were perfectly conserved between the AD and control networks, others showed substantial gene redistribution. Functional similarity analysis using gene ontology terms for biological process showed that 25 modules shared identical GO enrichment, and only 52 modules had at least 50% of similarity. However, several modules displayed distinct functional enrichments, indicating disease-specific expression patterns. Additionally, GO analysis across the entire network revealed a broader distribution of biological processes across modules in Alzheimer's disease, resembling a spreading phenomenon that further highlights reduced functional integration. Processes with modular dispersion in AD included neuronal development, neurodegeneration, axon guidance, and cognitive function. And processes with modular retraction included cellular transport, secretion control, and extracellular responses. This study provides insights into the systemic nature of the transcriptional landscape in Alzheimer's disease, considering not only linear relationship. Furthermore, this study may support the future development of a unified model for its causal mechanisms and diverse clinical manifestations. Thank you for hearing. Bye-bye.